forever grateful for because they gave me a fantastic start. So thank you, PBD. I have been mostly in the CRO world for most of my career, but heavily involved in what we call an FSP model, which is a functional strategic provider, which is basically can be two different things. One is basically you get the client's hardware, like laptops and stuff. For example, when I was working with MedImmune, we got a MedImmune laptop, and you log into the client systems and you basically interact with their team as if you are part of their team. There's really no delineation between a CRO resource versus a full-time resource. So that was a great, I would say, start into getting a view of from the side of things, because being on the CRO side of things, you don't necessarily understand nuances and all of the communication that goes behind the scenes with the pharma companies and biotech companies. So I have an extensive experience with regulatory submissions. I have had a few submissions under my belt. They're very few and far in between because this industry is a little bit different in that sense. Worked with GSK for their in their FSP model as well. I worked on their data standards conversion to CDISC standards, which basically led to their successful submission of Arzera back in 2014. It's also worked in the Cells FSP when I was with Lastar, which my work has contributions to their Veoza compound, led to their successful approval of Veoza in 2023, I believe. And of course, I'm currently with Neumora, where I have been directly involved in a lot of their entire portfolio for CNS studies, which includes major depressive disorder, schizophrenia, agitation due to Alzheimer's, and NLRP3, which is an obesity-specific drug. With biotechs, it's a different industry because you are basically following your passions. And part of the reason why I joined Neumora was because of my personal connection with CNS disease. My aunt has schizophrenia. My wife has major depressive disorder. And, I have other family members, uncles that have had Alzheimer's. They've passed away. So this is something that's really near and dear to my heart. That's one of the reasons why I joined a biotech company like Neumora, so we could make a difference in everyday people's lives. And mental disease is something that is difficult. It's a very difficult indication, or a difficult therapeutic area to be in. The indications like MDD, for example, they have a very high placebo response. So it is difficult that we have to navigate the data in a very nuanced way to ensure that we are presenting the right way, and also evaluating the data the right way, also analyzing the data in the right way. So this is, again, all, going back to all of the experience that I've gained from bigger companies, accumulating all of that and basically using all of those, basically an application of all of my experience and expertise into, a smaller company like Neumora. So that's basically my background. I think I talked a little bit too much, but
No, it's good. It's good. And there is a point I'll pick up that later, because you said something that was really, really interesting there. I'll pick up that in a bit. I guess, obviously, statistical programming, probably like a lot of people, or maybe it was just me. I think of statistical programming. I think of people with laptops just writing code, and that's the only thing that pops to my head. So I guess, what within drug development, what does a day-to-day programming role look like? What's involved? What is your day-to-day? What's that look like?
so what you just described as a statistical programmer, it's a bit of an antiquated theory, because yes, you are right. When I first started in the industry, we were considered as a very insular group, kind of pigeonholed, kind of heads down, code monkeys, if you want to call it. And my background myself, I have a degree in computer science and math, and so for me, it was where it was an industry where I could just code and I could just program. And even though I wanted to make a difference in patients' lives, I didn't know if this was one of those industries. But as the years have gone by and the role has evolved quite a bit, because statistical programming, we're kind of in the middle of data collection and analysis and reporting. So we have to not only make sense of the data that's being collected in the database, we also have to make sense of the analysis and reporting that we're doing at the very far end. So we're kind of the bridging gap between database design, CRF build, collection queries, all the way down to submissions to the regulatory body. So there has to be a lot of cross-functional collaboration that stats programming has to deal with. And even as a lead programmer, for example, if you're just a lead programmer on one particular study or two studies, you have to communicate with all the different stakeholders. For example, if we're talking about vendor data, you have to review the data transfer specifications. You have to talk to the vendors to talk to them about their data quality and the expectations of the data, how they're lined up compared to the specifications. You have to review the specifications, making sure that everything's correctly laid out in the document. And you also have to talk to, the statisticians and how they want to represent the data in the reporting, in the tables and the listings, and making sure all of those pieces are lined up so that at the very end, when we're analyzing this data or when we're reporting this data, it all kind of lines up. So that's where I think the role has evolved quite a bit. We're not just a fly on the wall listening to the conversations, but we're actually an active participant in the discussions. So I myself, I'm involved in a lot of the clinical study team meetings where I voice my opinion on whether a data point is viable or not, or how that one particular data point affects the endpoints for our particular trials. So it's very important for stats programmers to not just write the code, but also understand their code, the implications of the data that go into the code, and the output, how that implies. Also, the different stakeholders involved as well, your target audience, for example. My target audience can range anywhere from the executive leadership team all the way to, data managers or to clinical operations leads. So I have to basically morph or mutate my understanding or my presentation into the target audience, what they're looking for as well. So that's something that I think has been, again, a very big shift in programming in general. And we're not just, sitting in the back of the table, we're actually at the front of the table being part of the actual discussions.
Hmm. And I guess if we talk about the writing of the code, and when you're writing a code for a clinical trial, I guess the question being, why can't you just then reuse that for another trial or maybe in a different indication? Because you'd think it's a program, you write a program, surely if it works on one, it can work on the other. Why do you need to have specific codes for very different trials?
Right. That's a great question. So yes, I mean, the theory is that once you write a code, it's reproducible and you can reuse that, reuse, recycle. Um, but the reality is that it's a little different than that because every indication has different nuances in the data. For example, I mean, we're talking about, safety only, right? Like we're talking about adverse events, con meds, medical history, these sort of like standard vital signs, these sort of like standard safety responses, standard safety data that reporting, every study is reporting. But when you look at the different types of indication, I'm going to give you an example from the CNS field. So, for example, medical history, even within CNS, the medical history captured for major depressive disorder is very different than the medical history captured for agitation due to Alzheimer's because agitation due to Alzheimer's has a caregiver. So that caregiver themselves, there's that added variable right there that we have to now account for the caregiver's experience with the medical history for the particular subject or particular patient. Whereas an MDD, it's self-reported, and all of, these patients or these subjects relatively dependent from their evaluation or from having to need a caregiver, right? So there in itself, you can see that there is a lot of variability even within CNS. And again, for adverse events and concomitant medications, these sort of domains, again, we're taking a look at how, yes, adverse events, the AE term is loaded in, the start date, end date, you know, the severity, all that stuff is being recorded. But how that relates to the other domains is where the variability comes in. If there is a concomitant medication that affects, that creates an adverse event, how does that correlate with each other? And so there are different, again, data points where we have to, depending on the indication of the trial, depending on the therapeutic area of the trial, we have to then extract this information out of these standard domains to then present it in a way where it makes sense for that particular indication. And efficacy, of course, like within within oncology, there's so many different, indications. Within CNS, there are so many different indications. So like you have to tailor your efficacy and the way that you're analyzing the data that's written in the analysis plan in this tab, very different for every different indication. I think that's where you have to kind of change your code multiple times to fit that particular indication. Having said that, a lot of the data standards group are popping up. I think that's also a lesson learned from 20 years ago when I started in the industry to where we are right now. Is like data standards weren't really a thing until CDISC came in at like 2006, 2007 timeframe, which I was also involved in programming at that time. So from then on, the data standards have kind of took off going from CETA standards. And the reason why the data standardization needs to be in place, again, is the way to present it in a uniform manner to the regulatory bodies. So that there is a faster submission time, there is a faster decision from the regulatory bodies. And so again, all of that, again, also takes work. It's not just writing one program and saying, okay, I'm done. I can reuse this program for the next 10 studies. You also have to make sure that the regulatory bodies that you're submitting to, so the PMDA submission can look different from the FDA submission. It can look different from an EMA submission. So like submission, dependent material also is where you may not be able to reuse and recycle a particular program. You may have to write something additional for that particular submission. So these are some of the nuances that we have to think about whenever we're writing code and not just reusing code over and over again.
Yeah. And there's so many sort of acronyms in programming and in drug development, just in general, And the two big ones we always see when we're obviously trying to find programs, they need to have good experience within SDTM and ADAM. I guess, firstly, what are they? In basic terms, what are they? And yeah, what are the sort of differences between them, and how are you involved within that?
Yeah, so SDTM and ADAM, they're part of, and also C dash and other material from. They're basically coming from this group called CDISC, and CDISC was established actually here in Austin, where I'm based, back in the mid 2000s. And the sole purpose of, again, what I kind of touched on a little bit in my previous point was to standardize different study data, like different indications, different therapeutic areas. Like one particular therapeutic area, oncology, can have a very wide array of data collected and how it's presented, as well as how it's transformed into not just the data presenting, but also the data is collated together, for example. And back then it was kind of a wild west where, you would have 10 CRF pages and then to summarize it in one listing, for example. there it was a lot of inefficient work that's being done. So basically, see, this is what they did, and it's such a huge game changer in the industry. They standardized the data. So SDTM basically stands for Standardized Data Tabulation Model, which is basically standardizing all of the or collating all of the data from the database, like EDC bills, like various data. I mean, it doesn't matter whether it's a metadata wave or clinical, whatever database that you're using. You're basically collating all of that into definable domains. I mentioned adverse events, AE, conmeds is CM, exposure is EX, questionnaires is QS. So like they have all of these predefined variable names, variable labels, domain names, domain labels, where your data can be structured together. And it pools all the data from not just the EDC, but also the vendor data to then compile it all together in one nice big tabulated model, which then can be used in the analysis data and modeling datasets, which is ADAM, to then basically implement the SAP rules. So imputation rules in the SAP. There are, there's visa windowing in the SAP. All of that needs to be done in a separate level because STTM is just from a data collection and collation standpoint and standardization standpoint. When you go to ADAM, that's where all of the analysis steps are being put in. And that's where we basically in the old SAS days, ADAM dataset has to be one click away from producing a table. So to produce, for example, a CNS table, you have one prop mixed model or one prop to then transform your data into an MMRM model, which is then presented into the stats efficacy tables, right? So this standardization, I know it's a lot of acronyms and it's a lot of, I guess, forethought that needs to happen whenever you're starting up a study, when when you're even building a database or collecting different data points. But it serves a massive purpose in the in the machine of clinical research, because without it, just all over the place, and we would it would take the regulatory bodies, because there are so many different study submissions that are happening now compared to 20 years ago, so much more volume that the FDA wouldn't be able to get through all of it, or any PMDA or any regulatory body would be able to get through it without asking a lot of questions. Again, there's a lot of there can be a lot of ambiguity going from the database all the way to the reporting. So all of these standardized pieces remove the ambiguity as much as possible so that it can be all clearly explained, clearly traceable back to the original data, which is in the EDC database or the vendor data, and can be easily not just reproduced, not just produced, but also easily reviewed by the regulatory bodies.
One, obviously, there's obviously quite a lot of different programming languages that you would use and interact with every day. SAS obviously being the sort of industry standard for however so long, and obviously now R is becoming more and more prominent in the industry. I guess what are the differences between the different languages, but also then how would you use? Oh, sorry. How do you know when to use one for a certain part of your job, another for another part? Why not just have one language to use overall? So I guess that's my question.
Yeah, so that's one language for all is how it started in the industry, right? SAS being the very dominant uh,
Mm-hmm.
programming language to use for all clinical research, and SAS I think was a great start for us from a standardization perspective, and from on how to produce these data models, or how to produce these tables and formats, and or sorry, tables and listings and graphs. But however, SAS has limitations, and it is also very expensive. And so in an age where cost consciousness is very high, I think companies are, all searching for other alternatives like R and Python as well. And the the perfect and analogy is that you have to have different tools to do different things. So SAS is great at producing, a certain type of table, a certain type of graph. But if you want to take that in a different level, or if you are cost conscious about the amount of money that you're spending on a SaaS license, for example, the flexibility now that we have with the FDA being able to, or regulatory bodies are being open to submissions in other programming languages like R, is a very huge transition from how it was. So with R and with Python, there's a lot of data manipulation that you can do. There's also a lot of great visualizations that you can do. For example, I created an interactive dashboard for my executive leadership team, summarizing the primary and secondary endpoints, and it was very well received. And that's something that, with SaaS, I mean, of course, SaaS Via and all that stuff, that they're kind of catching up to it. But this is an easier and a lot more cost conscious way for people like me, or companies like mine, to be able to produce these results. And of course, R it's a community-based language, right? Like, so they have a lot of different community-based packages that you can reuse. Again, we're going back to reuse and recycle. You can have these packages that you can use to then come to your result a lot quicker without having to write the code yourself, without having to create the package yourself. Of course, you can do that, but it's going to take you a lot longer. So you can use the community fueled code or community fueled packages where you can get to your endpoint a lot faster. Uh, the other piece of it is, from a submission standpoint, I know that the FDA, and I'm really going on the FDA because I live in the U.S. and we do a lot of submissions. So the FDA had historically been only accepting SAS transport files, for example, which is, I think the deal that SAS made with the government back in, I don't know how long ago. So for electronic submissions. So the SAS transport files obviously have limitations. They have a 8 character variable length, 40 character variable label, and so on and so forth. So the FDA has evaluated other electronic submission data, like uh, like JSON, dataset JSON, where you are able to create XML data sets to where these limitations coming from SAS are now no longer applicable. So you are now free to create a data set where you can host text strings larger than 200 characters, for example. Before we'd have to break it down into 4 or 5 different variables. If it's like 1,000 character string, free text, you have to now break it down into 5 different variables. Now you don't have to do that anymore. There's a JSON, 1,000 character string. You can look at it in one go. So from that perspective as well, from a regulatory landscape, it's also evolving. And also these different types of submissions from a storage perspective, there are a lot less. Dataset JSON is can be a lot more compressible than a SAS transport file, for example. So from a space standpoint, storage standpoint as well, the regulatory bodies are exploring other opportunities, and I believe the pilot submission got approved for the dataset JSON, and now the version 1.1 is has gone through community review as well. I was also part of that, the work as well with CDISC, to evaluate and ensure that we have all the limitations identified and fixed in version 1.1 for dataset JSON submissions. So the landscape is changing, not just from the pharma side, the CRO side, but also from a regulatory side as well. So I think it's great to have all these different options. Again, is it going to translate into the Wild West that it was, 20 years ago? I don't think so, because we have these data standards, we have these, pre, or global clinical data standards groups at the various different pharma companies that are like really driving the need for standardizing all of the data. But how do we get to that data? Again, it's open season, right? I mean, it's different tool for different purposes.
Yeah. And you keep mentioning obviously about the regulatory and especially the FDA. I guess at what point, within clinical trials, do the FDA and the EMA, when do they actually see your work, and then also what is actually being presented to them?
Yeah, that's a great question. So when in the drug development cycle, you have the obviously you if let's say that funding is approved and like we have a green light on the study. Whenever we collect data from the sites themselves, that gets input into the database, which we call EDC, Electronic Data Capture System. And from the EDC data, there can be anywhere between months to years when the submission goes out to the FDA. So the work that's being done in between is, obviously a collection of all the data points, particular to the indication, particular to the trial, cleaning of all that data, standardizing that data into SDTM format, uh, creating the analysis data and modeling data sets to then now apply all of the statistical rules, the visible windowing, everything that I talked about earlier, and then create the tables, listings, and figures which are vital to presenting the data in a graphical or readable format. So all of that can take anywhere between a year to like multiple years, depending on how long the study is going. So at that point where when we are reaching close to an interim analysis or on a final database lock, that's when people, the pharma companies or investigators, they actually get to see, once you hit the lock, you see which patients are assigned to which arm. Meaning previously when things were all blinded, you don't know whether a subject is on a placebo or an active treatment. Only after database lock can individuals be unblinded to take the actual results, whether this drug is a success or not, basically. Of course, in the meantime, in the total duration of it, you're evaluating from a safety perspective as well. You're doing like blinded data summaries to take a look at the trajectory of the trial, how it's going. And then, there's like various committees like data monitoring committee, data safety monitoring boards that are involved, independent, as well as in-house committees that are involved, taking an in-depth look at the data. So once you get to that end lock, all of the safety piece has been evaluated at an ongoing piece. The efficacy piece gets evaluated at that point with the unblinded randomization codes. That's when you know when a study is successful or not. If a study is not successful, which is basically in layman's terms, like we talk about p-values, right? Like how it relates to the placebo. So the difference between the placebo and the active treatment, the p-value has to be less than or equal to 0.05. So that means it's statistically valid, efficacious result. At that point, that's when you decide, yeah, this drug is fit for submission. We have all of the, monitoring done. We have all of the collection done. We have all of the subjects cleaned, everything. That's when you start working on the submission pieces, which is, again, we're talking about reviewers' guides on how to review the data. We're talking about, again, standardizing the data even more into what we call Define XML, where we are collating all of the data from SDTM and from Atom into this Define XML wall. All of the coding, the control terminologies that are used in the trial, the value level metadata, all the values that are part of the data set, the particular variable, all is nicely collated in one web-based XML document, basically. And all the data underlying to it is also standardized and make sure that it's cleaned up. The FDA and also PMDA, EMA, they use a compliance tool called Pinnacle 21, which I'm sure you've heard of as well. We run that as well from the very beginning to the very end before the submissions to make sure that all the data that's expected in this particular set of data sets are compliant with the submission. Give you a small example from a trial design perspective. If the trial summary data is incorrect, and because we have to collect all of that and present that to the FDA, if the trial summary is missing or incorrect, that means that's an absolute rejection of the submission. So aligning all that up, it's not as very easy, even though we've been standardizing the data the whole way. We've been, using our analysis and modeling the right way the whole way. There still needs to be some sort of heavy lifting done at the very end before the submission. So that's where, the submission happens, and that's where the regulatory bodies actually get to see the work with all of the SDTM data, the add-on data, the defined XMLs, these reviewer guides, analysis report metadata, all of these different, BIMO listings we're talking about. It's, these are, I guess, like scientific listings, or that used to be called OSI, that they're reviewing to ensure that the safety and efficacy of the trial is all presented in that one package. And once they make sense of it all and they don't have any questions, then that's where the drug approval goes in. So it's a very long process.
No, it sounds it. It sounds it. And how do you do? And this probably would better suited to your work back in CROs. If you're running multiple trials all at the same time, always saying with fixed deadlines for the submissions, or just fixed deadlines in general, how do you balance all of that? Because it's just so much data and so many, and it's not like balancing, like a normal job. You make mistakes, it's millions and millions of pounds lost revenue. Submission fails, a lot of things. So how do you manage all of that? And maybe also, how many might you be running at one time, and how do you handle all those different data points?
Yeah. So you're absolutely right. I mean, it's all about prioritization and
Yeah.
organization. What the crux of it, what it all kind of boils down to is prioritization. So the whenever, yes, you're right, not just in CROs, but in pharma world as well. I'm part of a biotech where I'm responsible for 4 different compounds, multiple studies in those compounds, where I'm looking at all of the, day-to-day activities as well. And I have a team, of course, and I'm working with different individuals like stats, data management, all these different individuals. But the key piece is understanding where your priorities lie. For example, if there is a database lock coming up, or there's an interim analysis happening, these milestones are much more important than some of the early phase activities, like creating specifications or reviewing protocols, or reviewing SAP, for example. So hopefully all of those studies don't occur, all the timelines don't occur at the same time, so you can kind of actively manage where you can prioritize your time. Obviously the database locks takes the highest priority. The study startup takes a little bit lower priority. And in the middle, where the study is ongoing, there's data collection happening, there's, cleaning, queries, all that stuff. Yes, stats programming does have a vital role in that, but it is not an active day-to-day role in it. So there is like the life cycle of the study where you can kind of actively manage your time and your team to make sure that all of the activities are being done at an appropriate time. And I've had, multiple studies, like for example, with Neumora, we had 2 database locks for 2 studies happening at the same time for the MDD compound. So I had to manage my team. I had a West Coast, 2 programmers in the West Coast, and myself and another programmer were working East Coast hours. So we kind of divided up the work in saying, okay, the East Coast team will be working on this one particular study. The West Coast team will be working on one particular study, and we'll kind of come together, every day to ensure that we have alignment on expectations, we have alignment on our standards, and we have our alignment on how the data is presented, for example. So, this sort of like, the, I guess, leadership also comes into play, where you're actively managing ongoing studies or studies that are happening at the same time.
Mm-hmm. And obviously you mentioned you started off in a grad role in PPD as just I'm guessing just an entry level programmer, and now you've moved all the way up to sort of director. I guess how does the role evolve? From that entry level up to director, and are you still doing data like I call day to day data management programming? Are you still doing that? And yeah, and how does your role change and evolve over the 20
Yeah.
odd years that you've been doing this?
Yeah. So, from as an associate programmer, you're given tasks. You're saying, okay, somebody's giving you a task saying, hey, write these, you know, reconciliation listings programs, or write this one safety table, or write this one data set program, and you're given the work with the expectation that you're able to hit the timelines and expectations of good quality code, and talk to the people that you're associated with. If you're creating reconciliation listing programs, for example, that you talk to the data manager, understand their expectations and requirements for this particular reconciliation program, and then work accordingly. So it's kind of a trust exercise, as well as giving them a task to do. So that kind of evolves as you move up. You become you kind of take on a little bit more responsibility, from doing a task level to a study level. So like from a study level, there's a lot more happening at this at every level. You have data cleaning happening, you have data query resolution, you have you know tables being programmed for biostats, you have analysis data sets being created. So all of this takes a lot of work. So the lead programmer basically coordinates all of the work, hands out the tasks to the different, I guess the study level team members, like an associate programmer, for example, to be able to take on individual tasks. So that kind of amps up as you go on and on to from mid-level programmer to senior level programmer, and then you're expected to handle multiple studies at one point, because again, from the life cycle of a study, uh, you can kind of do the middle part of a study where not a lot is happening, and then focus really on the end parts or the beginning parts and the end parts of studies that are a little bit more intensive. So, and it kind of goes up to, in the manager level and the associate director level, you have to kind of learn to detach yourself a little bit more on the day-to-day work and day-to-day programming. Not saying that you completely alienate yourself from the day-to-day programming. That's not my style. I've always been a hands-on leader, where I understand exactly why, an efficacy table is taking entire week to program, or efficacy data set is taking an entire week to program. I understand that because I have done that myself, or I'm doing that myself on a lot of complex studies. So under having that understanding of what the ground level work is being done translates very well in your leadership style as well. Gaining empathy on people's struggles, understanding their weaknesses, and understanding their strengths. That's where a higher level manager or a director level can then direct their teams into making the most efficient most efficient solution for their team. And having said that, you also have to recognize where the weaknesses are and how to kind of prop up those individual teams or programmers into developing those skills as well. And that's making, again, a well-rounded programmer that can transition again to a higher level, like a lead programmer or, a manager. So that's, I guess, where all of the, as you go up in the levels, that's where things change a little bit. But one of the things I realized, and I myself was doing, is the detachment. Like when you're leading large scale teams, at one point I was leading 60 plus global programmers. It's a level of trust that you have to give to your teams. And if the trust is broken, or if the trust isn't there, then that means that relationship is never going to, just like personal relationships. You have a partner, you have to trust them to make sure that they do the right thing, and so exactly how that works in the professional level as well. You have to trust your teams, you have to trust your lead programmers to do the right thing, to follow up, to communicate with all these cross-functional team members that they're working directly with. So, and then coaching them, advising them if they when they're not. So that is an added layer of leadership at a director level where that's expected of you. And of course, the other side of things, which is the higher up, not just, going down, but like higher up, what is the expectation of you from the executive leadership team, for example? Like how you present, when you have all of this data floating around, how do you present that into an information so that the end user, which is the leadership team or head of clinical operations or the medical director, they can make sense of that data and they can use that to do their work better. I've created a lot of, I kind of was talking a little bit about like the R side of things. I created some dashboards for the leadership team where the leadership team can look at the endpoints, the primary and secondary endpoints, and pick out the filters for the patient population that they want to kind of drill down on and to see where the study is successful or not. So these are some of the things that at a director level that you have to think about, not just the people below, but also the people above and people around you, how you can help or how you can collaborate with everybody.
Hmm. Hmm. And I guess my last question is, you obviously, the work you do, and we always joke about, we call it the unloved child, is because biometrics and within drug development, whenever times are tough and it's time to cut money, biometrics is first one out the door. It's seen as like a cost center, but the
Yeah.
work you guys do is obviously so important, because without you, no trial would ever make any sense. You would just sort of have all this data and go, we hope it doesn't kill anyone, and that's basically what it is. So I guess with that, with the important work you do, but on the other side where it's not seen as important as say like clinical development or those types of things, why do you do it? Why not just go into like tech or finance, make a ton of money, and have quite a easy, not an easy life because there's no such thing as an easy life, but like an easier life than say what you have now? Why do it?
That I feel like is a bit of a personal motivation for a lot of people. For myself, again, with having a family history of illnesses ranging from oncology all the way to mental disorders, I had a personal motivation looking into this industry and seeing like these new therapeutics, or even not even just novel therapeutics, but like different mechanisms of delivery. I was once on a opioid dependence study back in 2007, 2008. That was a fantastic pathway. It was not a novel therapeutic. It was just the method of delivery. It was a subcutaneous implant under their forearm, and it would basically deliver the drug, which was already, clinically available in tablet form or whatever, but it would subcutaneously deliver, and it had massive effect. It had massive positive effect on the population. I mean, these are people that are, trying to get a second start in their life, like in halfway houses and stuff like that. So like that sort of impact I saw at a very early stage in my career. So that kind of really opened my eyes. That's why we're doing this. We're doing it to help people, not just make money for ourselves. And you mentioned like finance and other tech side of things. Yeah, you can make a ton of money very quickly. But in pharma world, it is what you're what the value you're bringing to society. I think that's where what motivates me, and that's what motivates a lot of people in this industry to be able to help people and be part of, the be part of society, like a productive part of society where you're not just doing it for yourself, but you're doing it for others as well.
Hmm. Yeah, perfect. Yeah, I think every person I've ever spoke to in biometrics and or in drug development anyway, I don't think anyone, except for one person, there was one person motivated by money, but most is about helping people. But you guys are very generous with your knowledge, and I really do enjoy talking to you guys. And Daniel, I really appreciate you spending some time talking to me, and thank you so much for your time.
Of course, thank you so much again for the opportunity. It's a pleasure talking to you.
Cheers, Daniel.